Hi everyone, my name is Alana Kalehua, and I hope you're having a great day. Today, we're going to be learning about molecular gradients that provide positional cues in the spinal cord and the head. Thank you so much. Let's get started. We have seen molecular gradients before being used in developmental neurobiology to indicate a cell's future function or destination. One example we've seen is pictured here, where certain molecular gradients in the gastrula will predict what this tissue will develop into. Molecular gradients here indicate that regions of the gastrula in yellow are low in nodal, BMP, and Wnt, and they will develop into brain tissues. The areas in the red are high in nodal, BMP, and Wnt, and they will develop into the more posterior tail of the animal. This is a great example to show us how molecular gradients can help us better understand the connections built in our developing brains and bodies. It is also a common mechanism of molecular gradients to act as chemoattractants or repulsants. This works to ensure that only the required resources are being recruited to optimize efficiency. And this efficiency is reflected in that we see these mechanisms in many creatures, showing that they are biologically and evolutionarily old. First, we're going to look at how molecular gradients act in the spinal cord, and specifically how they can contribute to the dorsal ventral axis. The spinal cord forms from the closure of the neural tube. The first image on the bottom here shows the development of the spinal cord after tube closure, where the floor plates and roof plates are established along with the notochord. These locations serve to secrete morphogens that will establish the dorsal ventral axis. The roof plate secretes BMP as the dorsalizing factor, and the floor plate secretes sonic hedgehog as the ventralizing factor. And we know these are the required morphogens thanks to experiments conducted by Thomas Jessel we've covered before. The gradient levels of BMP and sonic hedgehog meet in the middle of the system to create the sulcus limitans. And on the right, we can see how different molecular gradients can segment themselves into groups, which will become distinct cell types. Now we're going to continue to learn how a molecular gradient leads to differences in cells. By looking at the figure on the left, we see the blue represents sonic hedgehog being secreted from the notochord. As you travel away from the notochord, there's less sonic hedgehog, and the amount of sonic hedgehog surrounding a cell will activate specific transcription factors and genes to determine the neuronal subtype the cell will develop into and the future responsibilities it has. We can see that the ventral spinal cord produces at least 5 classes of neurons after the floor plate. The same process that the notochord uses with the sonic hedgehog to ventralize the spinal cord is also used by BMP from the roof plate to dorsalize the sensory horn. And both of these systems are represented together in the second image on the right, where we can see dorsal influence in blue and ventral influence in purple, and the relative colors indicate the gradients and the subsequent divisions into cell subtypes that will develop. The molecular gradients are determining the position where each cell type will develop. The head uses molecular gradients in a similar way as the spinal cord, even using sonic hedgehog as well to distinguish between different lobes of the brain during development. But we're going to highlight a different specific case of molecular gradients in the head to try and show the broad range of function molecular gradients can have. So in this example, instead of the gradient activating specific transcription factors in cells already present, we're going to learn about how molecular gradients help to form connections between separate regions of the brain. The brain uses molecular gradients to give positional cues as to where axons from the retina should be innervating the tectum. This system is responsible for sending the information our retinas see through the visual system so we can respond accordingly and safely. 2 spots close together in our retinal field will be mapped to close locations in the tectum, so it is important that axons are going to their correct destination to ensure we understand our surroundings correctly. The visual system uses effrin as ligands to bind with F receptors to match up axons with their final destinations. The nasal retina axons will innervate the anterior tectum, and the temporal axons will innervate the posterior tectum. The axons know that when they have reached their final destination once they have become saturated and repulsed by effrin. It is important to note here that effrin is a chemorepulsive molecule. If we look at this diagram here, we can see how this works. The nasal axons have very few F receptors, and so they can tolerate high densities of effrin. So they're able to make it all the way into the posterior tectum, but the temporal axons have many more F receptors and therefore cannot make it far into the tectum. The number of receptors an axon has will indicate how sensitive it is to a repulsive molecule, and when there are less receptors, the axon can travel further in the tectum before it gets overwhelmed. The first thing I'd like to add about the visual system using molecular gradients is that they continue to use their molecular gradient throughout the entire visual system. And this indicates it is a evolutionarily old system and successful to have remained in place for so long. And it makes sense why a system might develop this way, because connecting all the input along with the same messages can help to ensure that things don't get lost in translation along the way. But this also means that any deficits within this molecular gradient system would likely lead to significant visual problems, as the system would be interrupted along every step and not just in one brain region. And while on the previous slide, we only looked at one efferent and F receptor, there are many. Multiple molecules within the same family exist and are used in order to increase the precision of visual information. The second image on the right here shows how multiple molecules can work together to indicate exactly where stimulus is in the visual field. Another important aspect of efferents is that they are bifunctional, and this means that they can have multiple different effects. It has been shown that efferent can have repellent and attractive effects, and the difference in effect is concentration dependent. So low concentrations of ligand will be attractive and high concentrations will be repulsive. And this is important because the brain is using the same molecule to do two different tasks, even opposite tasks. And so it's important to recognize that one molecule may be very versatile and could be doing more than only what we initially observe. So thank you so much for listening to me talk about how molecular gradients can provide positional cues in the spinal cord and the head. We learned that they work in the spinal cord by secreting a gradient to activate transcription factors and genes to tell a cell what it needs to become, and also how molecular gradients can work to give axons their final destination cues. Thank you so much for listening. Have a great day.